Evening, everyone. My name is Meghna Saha, and I am a student at Naperville Central High School. Today, I'm presenting my research project conducted under the mentorship of Dr. Jingo Agay, titled Impact of Alcoholic Cardiomyopathy on Mitochondrial Structure, Myosin ATPase Activity, and Cardiac Function in rat models. So before we go into detail, let's go over our agenda today. So first, I'll be talking about our background. So what is alcoholic cardiomyopathy? Then I'll be going through what our research hypothesis was, and then our desired outcomes. After that, I will go over our method and how we conducted our research. Then I will go on to analysis and results and analyze the data that we have extracted. Then, right before our conclusion, I will address some continued research, some research that we would like to continue, and then our conclusion, wrapping up everything. So, what is alcoholic cardiomyopathy, or ACM? ACM is a cardiac disease caused by chronic heavy drinking affecting 1 to 2% of heavy alcohol users and accounting for approximately 21 to 36% of non-ischemic cardiomyopathies. The disease impacts the heart's mass and volume and leads to impaired cardiac function, mitochondrial damage, oxidative stress, and altered calcium homeostasis. So, in this study, we really just focused on mitochondrial damage and oxidative stress and how this affects the ATPase, right? So, our research hypothesis is that chronic alcohol consumption causes significant mitochondrial damage in cardiac tissue, leading to reduced ATP production. To prove out the above hypothesis, our research focused on comparing healthy hearts with those affected by ACM in rat models, focusing on mitochondrial structure and myosin ATPase activity. So here is an overview of our method. So to test our hypothesis, we compared heart tissue from healthy rats with those affected by ACM. As you can see on the left here, we had two groups: our control, which had a healthy diet, thus normal heart, and our alcoholic rich diet group, which, as you can see on the right arrow, that had a heart with dilated cardiomyopathy, or ACM. Then on the right, you can see a much more detailed version of our method. So first, we take samples from both groups, and we do two things with them. We have two parts to our method. Our first part is that we take images of the mitochondria samples from each group and analyze for area and density variation, and then compare that. Our second part of the method was that we analyze and compare the myosin ATPase rate from the samples from each group. Then, by analyzing and comparing what is already known about the effects of alcohol cardiomyopathy to what we collected in our study, we were able to confirm the damage that alcohol does to the heart. So, after analyzing the photos of samples using ImageJ, our research revealed significant swelling of mitochondria in the alcohol-rich diet group. The increased area and electron lucency, aka the variation in density, indicated substantial mitochondrial damage due to oxidative stress, which we can see in the following graphs here. When comparing both the alcohol-rich diet group and the control group, we can see that there is a higher variation in the density and area of the mitochondria in the alcohol-rich diet compared to the control. This indicates that the mitochondria are swelling and becoming damaged due to alcohol consumption. Since mitochondria are what form ATP, damaged mitochondria means a reduction in ATP production. So, for the sample extraction, just as a part of the sample extraction, actually, I was able to go to my mentor's lab and was able to perform the sample extraction. So before I start on what exactly the extraction was, Let me first define what myosin is. So, myosin is essential for muscle contraction, and its functions can be impaired by alcohol induced mitochondrial damage. To understand how it is impaired, we first extracted and purified myosin from the rat heart tissues using a series of centrifusion steps, followed by the SDS gel electrophoresis to verify the quality of our extractions. As you can see, the SDS page gel on the right, along with a picture of one of the samples. Then we conducted an assay, which is a process of analyzing a substance to determine its composition or quality. We use this assay to measure the ATPase activity using a colorimetric method, tracking phosphate liberation over time. The ATPase rate was significantly lower in the alcohol-rich diet group compared to the control group, indicating that alcohol impairs the function of myosin ATPase, which in turn affects muscle contraction efficiency. As you can see here, the alcohol-rich diet group has a lower myosin ATPase rate than the control group. So, a comparison of these results. Here you can see an overall comparison between the alcohol-rich diet group and the control group, showing that the mitochondrial area and the mitochondrial leucine, or the variation, are higher, and the myosin ATPase is lower in the alcohol-rich diet group. So continued research. Our findings open up several avenues for further research. We plan to explore the effects of alcohol on calcium regulation within cardiac cells and investigate how calcium homeostasis disruption leads to mitochondrial damage in the heart. Additionally, we aim to examine the impact of alcohol-induced muscle disorders in the brain on cardiac health and identify potential therapeutic targets to mitigate alcohol's harmful effects on the heart and overall health. In conclusion, our study highlights the detrimental effects of alcohol on mitochondrial structure and function, as well as on myosin ATPase activity in cardiac tissue, like how we observed mitochondria with higher density variation in the alcohol-rich diet group, indicating significant damage. Mitochondrial damage that led to reduced ATP production, impacting ATPase activity. These findings underscore the significant role of mitochondrial damage and oxidative stress in the development of alcoholic cardiomyopathy. And finally, we come to the end of our presentation. So some acknowledgements. So first, I would like to thank my mentor, Dr. Jingwa Gay, for allowing me the opportunity to learn so much from her and to be able to go and work in the lab with her. And it was really an amazing experience, and I truly enjoyed my experience with her. And if I were to recommend her later, uh, in any other project or anything, I would obviously recommend her. And then obviously Talaria Summer Institute 2024 for allowing me the opportunity to actually be able to do research, because I never really had any opportunity to do this, and this is my first time. And yeah, it's like a really memorable moment for me. And then obviously Penn State, Dr. Christopher Yengo's lab and Department of Cellular and Molecular Physiology for faculty support, and obviously all of you who are listening to my presentation. So I look forward to any questions you may have. Thank you.